Thanks a lot. I was asked to step in for Edith Hurd, who could not attend the conference. And don't worry, I'm not going to speak about pediatric oncology again, but focus on the next step for translational research. And I just came up with a couple of questions that I thought we could address in the further panel discussion, because we covered a lot of aspects on translational research already in the past 2 days, a lot of challenges, a lot of opportunities, a lot of needs. But I thought about what are the remaining questions that I can't answer so easily, and maybe it's worthwhile to discuss them. So the first question coming to my mind was how to assess success of translational research. What are the key performance indicators? So if we judge translational research and translational research output in the same way as we do currently judge basic research, we might not justify this appropriately, because of course it's sexy to make discoveries, to publish them in Science and Nature, but that doesn't mean the translational process has successfully been made. So what are the key performance indicators that we would go for in assessing the success of translational research? And we had a lot of discussions on this also in the German Cancer Consortium. How would be a good incentive also for young researchers that they go the lengthy process of validation, of bringing for example, diagnostics to the clinic, even if this is not a sexy new discovery, but just a very long and maybe not so sexy process. So the second question I asked myself was, how precise do we want to be on the molecular level? I'm personally a fan of single cell level technologies, but is that really needed? We still need the proof of concept that we can get to new drug targets and a new level of drug targets maybe when we go to the single cell level. And at least to my knowledge, this proof of concept has not been made yet. So we might consider this to incorporate this question also in future clinical trials. And then discussing with basic researchers, I find that their knowledge of how to bring a new biomarker that they have identified in their labs, how to bring this to the bedside, that the knowledge about the process is in some of them not very strong. So not all of them know about the medical diagnostics regulation and what kind of accreditation processes are meanwhile needed before you can base a clinical decision on a new biomarker, and how lengthy this accreditation process can be and how to approach it. Also, how do we ensure that we continuously incorporate new technological developments, and when do we consider these to be ready for personalized medicine applications? Do we want all kinds of developments? How do we assess their value for the patients? And if we have a look at that on the global level, we will definitely not be able to finance single-cell molecular diagnostics worldwide for every cancer patient. So how do we deconvolute these high-tech data? Can we maybe dream of using just immunohistochemistry that in every country of the world is there? If we train for example, spatial transcriptomics data with AI and extract the meaningful information and extrapolate that on normal H&E stains, and can we do predictions then from there? And by this approach, transfer, just as an example, transfer our high-tech new knowledge more globally to countries that have more limited resources. And then in the field, of course, of drug and ATMP development, how do we want to approach this? How much should be done in the academic setting versus industry? Would there be new ways, new partnerships, how to explore this path together, which could be more efficient? How is our idea what this should be in the future? And then in respect to patient representatives, at least in Germany, we feel that many of our patient representatives are meanwhile very familiar in evaluating clinical trial designs and judging on these, but this might still be different, at least in a fraction of the patient representatives if we are talking about wet lab projects. So how do we train them to assess biological questions and research proposals in that field? And finally, we heard numbers about basic research, and there is, thank God, a lot of funding going into basic research still. On the other hand, I think if we don't fund basic research sufficiently, at some point we won't have anything to translate anymore, because the major part of discoveries are made in basic research. And it is our task as clinicians, as clinical scientists, to provide the pathway how to take up these discoveries from basic research and deliver it to the bedside. And this, for me, is the most important discussion in the next steps for translational research. Thanks a lot.